right, good afternoon, my micro students. Welcome to Chapter 8. Today we're going to discuss some topics that cover microbial genetics and genetic engineering. The next slide describes the outline for this chapter. We're going to cover 8.1, 2, 4, 5, and 6, but we will skip 8.3. So first and foremost, 8.1 gives you an introduction to genetics and genes. Genetics, by definition, is the study of genetic material, which is DNA, or deoxyribonucleic acid. Deoxyribonucleic acid. So that's where DNA gets its name. This field of study encompasses a few different things. It encompasses the structure and the function of genetic material, or DNA. It discusses heredity, which is how DNA is passed from parent to offspring. It discusses how DNA is expressed, which means how DNA physically manifests into traits. For example, your eye color is the color that it is because you have DNA that corresponds with the various proteins that give your eye that pigment. And also it encompasses the understanding of how this material of DNA changes. For example, mutations. Before we get started, there are some general terms we need to be familiar with. They are listed here on the PowerPoint slide, and we're going to talk a little bit about each one pretty briefly. So starting with genome. Genome is the sum of all the genetic material of an organism. And DNA is going to be organized into chromosomes or plasmids. So if we look at the eukaryotic cell, so as a reminder, these are cells that have a nucleus, as you can see here. Comparatively, bacteria does not have a nucleus, neither do viruses. But these cells that are eukaryotes have a nucleus, and their genome is organized inside of the nucleus as linear bars that look like X's in this picture, but if you can imagine, half of that X is a linear bar. If you've taken biology with me, you know if they're X's, they're replicated chromosomes, but that's kind of irrelevant for our purposes in micro. But our eukaryotic cell has a linear DNA that is organized, a linear chromosome that is organized inside the nucleus. Comparatively, bacteria, we see a circular loop of a chromosome. So just compare for a second. Linear bars here compared to the circular loop. Also, bacteria will oftentimes have plasmids. As you can see in this picture, they're much smaller than the main chromosome. They'll have basically fluff DNA. It's like extra stuff. Now that doesn't mean it doesn't serve a purpose. It can have really awesome traits that it gives to the bacteria. We'll cover that later. And then viruses, as you can see, has DNA or RNA that it's going to be packaged inside of the capsid. But again, the genome, by definition, is the genetic material of an organism. So if I were to analyze all of your DNA, that would be your genome. So let's compare chromosome and plasmid. A chromosome is a distinct cellular structure composed of neatly packaged DNA. As we described previously, eukaryotic chromosomes are linear. So what we're seeing here is a slightly larger picture of the eukaryotic cell. We see the chromosomes within the nucleus. And if we were to analyze this chromosome on a more microscopic level, it is made up of course, DNA, as we know, and we've seen DNA structure before. So this DNA, which is linear, is going to wrap around these little purple balls, and these little purple balls represent proteins called histones. So as you can see, it's going to wrap around them, and that wrapping will eventually lead to what we call supercoiling to formulate those chromosomes. So basically what you're seeing here in the chromosome is raveled up DNA. And again, it's linear. If we look at bacterial chromosomes and plasmids, on the other hand, they are packaged by the same means of histone-like proteins, but the chromosome is circular. So instead of what we saw in the previous image, a chromosome being linear like this, and I'll draw the other side, this chromosome is condensed around histone proteins just like we saw with eukaryotes, but it forms a circular loop. So this is very long, but if we were to stretch this out, you would see that it would essentially have this shape. So you can almost think about this as like a really long rubber band. And if you were to put it inside of a little compartment, it would fold over on itself to fit in that compartment. So that's analogous to what you're seeing here. This is a single circular loop of DNA here in blue. It's just kind of packaged all together and clumped into one spot inside of this bacterial cell. So this circular chromosome is what contains vital genes, meaning it keeps this cell alive. It has to have those genes. The plasmids, on the other hand, in red, they contain genes that are for other stuff, things that aren't needed for survival, things that are extra. Like, for example, possibly bacterial antibiotic resistance genes are found on plasmids. They're not necessary for survival, but it does give it some influential traits. Our next definition are genes. Genes are segments of DNA on a chromosome that codes for RNA, which will eventually be converted into a protein. We'll talk about protein synthesis later on, but for right now, what I want you to know is that a segment of a chromosome, and again, we see a linear chromosome, so it must be from a eukaryotic cell in this picture. But if we were to unravel it, it is made up of DNA, and a segment of that DNA would ultimately code into a protein product. And there's lots of steps that go on in between, but we'll talk about that later. So a gene codes for a protein, and thus it yields some kind of trait. So for example, if you have a lot of special types of melanin for hazel eyes, you will produce proteins that will be seen as that color, and so on. That brings us to a couple more terms, genotype and phenotype. So our genotype is the summation of the different types of genes that we have, whereas phenotype is how those genes express. So to compare, if we look at this in the form of like a human, which is probably easier for us to understand initially, the genotype we have is going to be the collection of genes we have. The phenotype is how it's expressed. So if I were to analyze the genotype for this person, they would have genes that code for straight hair. The fact that their hair is straight is their phenotype, because that's how the genes are physically manifesting, how we see it. Someone who has curly hair will have a different genotype than the person that has the straight hair, because the curly haired person will have genes that code for curly hair. So again, genotype is the type of genes you have. Do you have genes for brown eyes or blue eyes? Do you have genes for curly hair or non-curly hair? Do you have genes for short fingers or long fingers? The list can go on and on. But the phenotype is how that genotype expresses itself. So this phenotype can also vary with the cell environment, because we can regulate genes and turn them on and off. But let's look at this picture here. It describes the difference between genotype and phenotype just in a different organism, here in a bacterial cell. So genotypes would be found on chromosomes and plasmids. What sequences of A's, T's, G's, and C's do you have? Whereas the phenotypes would be the physical manifestations. So for example, the capsule. If it has a capsule, it must have a gene that codes for a capsule. If there is a flagellum, then there must be genes that code for said flagellum. If it's able to produce conjugation pili, then there must be a gene that codes for that. So genotypes are the type of genes you have. Phenotype is how it physically manifests into some kind of structure or function. All right, moving on to DNA structure. So DNA structure is universal pretty much, except for some viruses. We've talked about DNA a little bit in the past, so y'all probably are familiar with a lot of the things we're about to talk about, especially since you've probably had seen this in A&P, and you've definitely seen this as a biology student. But DNA is going to be a little bit different sometimes in viruses because they can do weird things like we talked about in chapter 5. For example, they can have single-stranded DNA, which is not something we ever talked about in any prerequisite course. So anyway, we're going to talk about good old regular DNA because we've moved on from viruses, but that was in chapter 5. So DNA is a polymer of nucleotides. Polymer meaning many units. Poly, many, mer, units. So nucleotides are going to contain 3 things: a phosphate group, a sugar, and one of 4 nitrogenous bases. So if I were to show you that in picture format, this is what I would show you. We have our phosphate group, we have the sugar, and we have one of 4 nitrogenous bases that would be here. The 4 possibilities that this could be are adenine, thymine, guanine, and cytosine, and we abbreviate those as A, T, G, and C. And so again, DNA is a polymer of nucleotides, meaning it's many of these. So next we're going to describe how those are linked together. So these nucleotides are linked linearly via covalent bonds between the sugars and phosphates of the various nucleotides. So if we look at this linear combination of 1, 2, 3, 4, 5, 6 nucleotides, you can see there's a phosphate group, a sugar, a nitrogenous base. In this case it's adenine. Phosphate group, sugar, nitrogenous base. In this case it's guanine. Phosphate group, sugar, nitrogenous base. 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 So we have a grand total of 1, 2, 3, 4, 5, 6 nucleotides that are linked covalently between the sugar of the first nucleotide and the phosphate group of the next nucleotide. Right, sugar of the second nucleotide, phosphate group of the next nucleotide, and so on. So that creates what we call the sugar phosphate backbone. I'm just going to kind of draw a rough schematic here. But that right here, this red line that is poorly drawn, my apologies, is the sugar phosphate backbone, because the sugars and the phosphates are essentially the backbone of this linear structure, and the bases hang off of that. And the bases are here, the A's, T's, G's, and C's. Well, that's not all, because DNA is not a single linear structure. It is actually a double linear structure that's twisted like that's twisted into a helical shape. So the second strand of DNA is going to be chemically bound to the first one in a specific manner. Adenines always bind to thymines, and guanines always bind to cytosines. This is the law of complementary base pairing. So what y'all saw previously in this image was a single linear strand of DNA. So essentially this is a single linear strand of DNA right here. The other linear strand is on the right. And you can see that the nitrogenous bases that are hanging off on the inside pair up in a specific way. A pairs to T, G pairs to C. See, A pairs to T, G and C, G and C, A and T. So ultimately then, if we simplify this, it kind of looks like a ladder, two-dimensionally. So this picture here, the first one, is the same thing as this one. They're just simplifying it. They did not show the sugars and phosphates. They represented them as these dark blue-green lines on the left and the right, and the nitrogenous bases are hanging off in the inside. Again, matching accordingly to the law of complementary base pairing, where C pairs to G and A pairs to T. But that's not all. So in reality, DNA is not this flat ladder shape. It is going to be twisted like this to form a helical pattern. Hence why the DNA structure is oftentimes referenced as a double helix, because it's two strands and it has a helical shape. So you can almost think of it like a spiral staircase. Like a spiral staircase. So, if we were to analyze that in humans and bacteria, again, we have our eukaryotic chromosome that sort of looks like an X. And if we were to unravel it, now you see why it has that helical shape that is got two strands. Same thing with bacteria, it just forms a loop. Alright, this is a picture that comes from your text, so I just wanted to go over that. It's the same things we've already talked about. I just want to make sure that you understand it in reference to the pictures from the book as well. So what this is showing is phosphate group, sugar, phosphate group, sugar, phosphate group, sugar, phosphate group, sugar, so on. So that's our sugar phosphate backbone here of strand number 1. Alright, S1. Strand 2 on the other side, we see the same thing. Sugar, phosphate group, sugar, phosphate group, sugar, phosphate group, sugar, phosphate group, all the way down. And then in between, we have our nitrogenous bases. And as we know, they're always pair up, where guanine or G pairs with C, and A always pairs with T. And so if you look all the way down, you will see that, in fact, that we do see that pattern where A pairs with T and G pairs with C. There's a little bit of new information here. I think I'm only going to cover one word because we're going to talk about this 5' 3' stuff later. But you might be wondering, okay, what's holding this together? And the answer is hydrogen bonds. These dotted lines represent hydrogen bonds that are holding together strand number 1 and strand number 2. Those bonds are going to be in between the nitrogenous bases. Alright, let's watch a video.
The structure of DNA aids in the speed and accuracy of replication. Double-stranded DNA is a polymer of two strands of nucleotides, which are hydrogen bonded to each other to form a double helix. Nucleotides are molecules that consist of a deoxyribose sugar, a phosphate, and one of 4 nitrogenous bases. The phosphodiester backbones consist of alternating sugar and phosphate groups. The nitrogenous bases include cytosine, thymine, adenine, and guanine. Cytosine forms 3 hydrogen bonds with guanine, and thymine forms 2 hydrogen bonds with adenine. This is referred to as complementary base pairing. The double helix will have one strand oriented in a 5' to 3' direction relative to the hydroxyl group of the deoxyribose sugar, and the other strand oriented in a 3' to 5' direction. This shows the anti-parallel nature of the DNA strands.
Alright, guys. So the thing that they just mentioned that I told you I was going to hold off for a second was that 5', 3' stuff. So it is in this previous picture, but I don't like the way that I wanted a different picture to show it to you. So I wanted this picture. So what do they mean by that? So if we look at the sugar of any of these nucleotides, we can count them and name them 1', 2', 3', 4', and 5'. The way that we do that is to start at the oxygen and then go clockwise, which is to the right. This intersection of these three lines here would be carbon number 1. You might be asking, why isn't C listed there? And that's because it's a simplified picture. Oftentimes in biology and chemistry and organic chemistry, we simplify things because they're assumed. And so in organic chemistry, anytime you have 2 lines or 3 lines, however many, intersecting in a chemical diagram, it's assumed that that is a carbon. And then for those y'all who took biology with me, y'all know that carbon can bind to 4 things. So in this scenario, since it's only bound to 2 things that we can see, 2 other carbons, it's also bound to 2 hydrogens. They just don't draw them in. Now, do you need to know all these rules? No, but you do need to know enough to where you understand what I'm referring to when I talk about 5', 3' and directionality. So again, we start at the oxygen and we count clockwise, 1, 2, 3, 4, 5, and you can see the little tick marks behind each one. Those are the symbol for prime. And if we look at this one, 1, 2, 3, 4, 5, 1, 2, 3, 4, 5, 1, 2, 3, 4, 5. All right, so again, starting at the oxygen, you count the carbons clockwise. So if we do that now on the other side, kind of a weird, doesn't look the same, does it? No, it doesn't. If we flip this over, it would. But the reason for that is because of the anti-parallel nature of DNA, where the complementary bases are A and T, G and C. So if we look here, let's start from the bottom. If we look at this very first sugar, now you got to, again, remember, look, it's upside down compared to this one, hence the anti-parallel nature. So if I'm counting the carbons, I had to start at oxygen and go clockwise. 1, 2, 3, 4, 5. Same thing here. 1, 2, 3, 4, 5. Same thing here. 1, 2, 3, 4, 5. Same thing for the last one. 1, 2, 3, 4, 5. So now if I analyze this first strand, which of the carbons will I come across first? 5, 4, 3, 5, 4, 3, 5, 4, 3, 5, 4, 3. And for that reason, this strand is called the 5 to 3' directional strand. Now what about this one? If I'm going top to bottom, I come into contact, or when I look at it, of the first carbon, which is 3', 4, 5, 3, 4, 5, 3, 4, 5, 3, 4, 5. And for that reason, we call the second strand the 3 to 5' strand. And it is going to run in the opposite direction as that first strand. So in DNA, we will always have 2 strands, except for viruses sometimes. There will be 1 strand in the 5 to 3' direction, and the second strand will be in the 3 to 5' direction. And so that's what they meant by that 5, 3, 3' business in the previous image in that video. So the amount of that I really need you to know is if I were to give you a DNA strand, you should be able to tell me what the other DNA strand reads. So we can shorthand what we've seen so far even further by strictly only writing the A's, T's, G's, and C's, because the rest of it's assumed. So if I were to give you the DNA strand, 5', 5'N begins with A, and then there's G, T, C, T, A, G, C, A, and then we end with a 3', because this is the 5 to 3' direction. And I asked you to give me the other strand, you would tell me 3', T, C, A, G, A, T, C, G, T, 5'. So together, the first strand, I just copied and pasted here to here. And then the strand that you would have told me was complementary, I put at the bottom, and it's color-coded as you can see. You can see the top strand's going 5 to 3, the other strand's going 3 to 5. If this were to have said 5' here and 3' there, that would have been wrong. You always have to have one that's in the 5 to 3, and the other one must be in the 3 to 5. And I just realized I forgot my little tick mark. So I'm going to fix that. Okay, there, now I'm happy. So you always should know one goes in 5 to 3, the other goes in 3 to 5. And how did I know what the letters were? Again, complementary base pairing. This A was known, and it must pair to T. G must pair to C. T must pair to A. C must pair to G. T must pair to A must pair to T, and so on and so forth. All right, so the next part of our text reviews replication. I'm not going to cover replication because it's not part of our state standards, and it probably was covered in another class. I'm not sure if A&P does that, but we definitely do it in biology, and it's really not relevant for us to talk about it for micro. So if you want to look at the video that have embedded here, you can play it, or you can use the link if you want to just have a really brief overview or review, depending on if you've seen it before, but we're not going to actually cover it in the PowerPoint, and you will not be tested over it either. However, it is more important for us to talk a little bit about transcription and translation. These are the two steps that are involved in protein synthesis. So protein synthesis is how we make proteins, and we call this flow, this process that genetic information moves through, the central dogma of molecular biology. What is a central dogma? What do we mean by that? Central dogma, it just means the big idea. The big idea behind molecular biology and the flow of genetic information is that we begin with DNA. That DNA is converted into RNA via a process called transcription, and then that RNA is translated into protein via the second process called translation. So let's start with transcription. So again, we start with DNA, and it's going to be converted into RNA. So before we get into the details of how transcription works, I want us to review a little bit of how DNA and RNA are structured. We've already went through today the structure of DNA. I drew in or added in a picture here just as a reminder and a comparison. But RNA, which stands for ribonucleic acid instead of deoxyribonucleic acid, RNA is single-stranded. So as you can see in this picture here, it is single-stranded rather than double-stranded. It contains uracil instead of thymine. So if you look at the possible nitrogenous bases listed for RNA, we see A, G, C, but no T. No T. Instead, we see uracil. There is no uracil in DNA. There's thymines in DNA, but no thymine in RNA. It has uracil. And then our last difference between RNA and DNA is the sugar. So we know that both of these two molecules, DNA and RNA, are polymers of nucleotides. And again, y'all told me, or we covered, that nucleotides have three things: phosphate group, sugar, and nitrogenous base. The sugar varies. In DNA, we have a deoxyribose sugar, hence the name, once again, deoxyribonucleic acid. In RNA, we have a ribose sugar, hence the name ribonucleic acid. What's the difference? This right here. So the sugar is almost identical, except in deoxyribose sugar, it's lacking an oxygen that we see in the ribose sugar of RNA. Also, hence the name, deoxyribose. It's a ribose sugar, essentially, that's missing or lacking an oxygen. This sugar has one less oxygen here compared to ribose. That's why it's called deoxyribose. All right, next, RNA comes in three varieties: mRNA, tRNA, and rRNA. mRNA stands for messenger. And we say messenger RNA because it provides a message, a copy of a message from DNA to eventually a ribosome that will make it into a protein. So it's the messenger here. You can think of it like DNA is the blueprint of how to make proteins, but it's not in the right language. We need a translator. So this mRNA is the messenger that converts the message of DNA into a readable message of RNA that can be read by our ribosome. So this part here, this conversion of DNA to RNA, messenger RNA specifically, is relevant for transcription. The other two are tRNAs and rRNAs, which stands for transfer and ribosomal RNA respectively, are relevant for translation. So in summary, I included this chart to give you the various types of RNA classifications and their characteristics and key functions. I'm not going to read this to you. It's basically covering mostly what I've already said. There might be a few other small details, but this will help summarize things really nicely. So now let's talk about the process of transcription. So again, transcription is the process of converting DNA into mRNA. It's going to be it's going to have 3 steps: initiation, elongation, and termination. So in initiation, there is an enzyme called RNA polymerase, which will bind to a template strand of DNA, and it will read the sequence. It will know where to start reading the sequence because it will find this little area here called a promoter. That little area is called a promoter. It's basically a flag that tells RNA polymerase where to begin reading DNA. So once RNA polymerase has found that promoter sequence, we will now go into the elongation phase, where RNA polymerase will begin reading each of the nucleotides on DNA and then bringing in an RNA nucleotide that is complementary to what was on the DNA template. And then finally, termination. We will eventually reach, the yellow part here is representing a stop sequence, and that sequence will tell the RNA polymerase that we're done, and that will terminate this transcriptional process. And so we'll be left with this mRNA transcript in, I guess that's an orangey color. All right, let's watch this video.
During transcription, the DNA in the gene is used as a template to make a messenger RNA strand with the help of the enzyme RNA polymerase. This process occurs in 3 stages: initiation, elongation, and termination. During initiation, the promoter region of the gene functions as a recognition site for RNA polymerase to bind. This is where the majority of gene expression is controlled, by either permitting or blocking access to this site by the RNA polymerase. Binding causes the DNA double helix to unwind and open. Then during elongation, the RNA polymerase slides along the template DNA strand. As the complementary bases pair up, the RNA polymerase links nucleotides to the 3' end of the growing RNA molecule. Once the RNA polymerase reaches the terminator portion of the gene, the messenger RNA transcript is complete, and the RNA polymerase, the DNA strand, and the messenger RNA transcript dissociate from each other.
All right, heading back over to our PowerPoint. Finally, I'm just going to show you a quick example. This is showing you our DNA molecule here. One of the strands of DNA will serve as the template, and as you can see, the DNA template reads TAC, TAG, AGC, ATT. So when RNA polymerase finds the promoter and begins reading that template DNA sequence, it will add in RNA nucleotides. So what's complementary to T? A. What's complementary to A? And in this case, it's U, not T, because we're making RNA. And remember, RNA does not contain thymine. It has uracil instead. So then we see a C pairs with G. T pairs with A. A, again, pairs with U, not T, in this scenario because we're making RNA. G pairs with C, A pairs with U, and if we kept going, we would finish out this transcript. So now, of course, this is just a very short example. In reality, the average mRNA is going to consist of like 1,200 bases, not just like the, I don't know, like 10 or 12 that we have here. Okay, so now we're shifting gears. We already discussed transcription, which is taking DNA and converting it to RNA. We're going to move on now to the second process, which is translation, where we take that mRNA and now we're going to translate it into a protein. So in this process, it's going to associate with a specific organelle called a ribosome. So the mRNA that we just made will find a ribosome, and the ribosome will read the mRNA, just like it's a book, in 3 bases at a time, which are called codons. So if we see this mRNA transcript here, it doesn't show the ribosome, but that's okay. I'm just trying to show you codons right now. And if we were to count the first 3 nucleotides, A, U, and G, this would be codon number 1. The second triplet would be codon number 2. The third triplet would be codon number 3, 4, 5, 6, and 7. Alright, so the ribosome is going to read those codons on mRNA 3 at a time, and that is what a codon is called. Now, when each triplet is read, the ribosome will bring in a transfer RNA, or tRNA, that has an anticodon on it. An anticodon is 3 bases that are complementary to the mRNA's codon. And it will continue to do that 3 nucleotides at a time until translation is complete. Now, I know that might sound a little confusing. Let's apply some pictures to it. I want to first talk about the ribosome. I feel like it's important for us to know a little bit about its structure to understand how it works. So here we have the ribosome. It's made up of a large and small subunit, and whenever they nestle themselves together on a piece of mRNA, this is showing where the mRNA would be, it kind of sandwiches it. The sandwich would then have exposed areas here, here, and here, APE, APE. Those sites are called the A site, the P site, and the E site. Let's also talk about the structure of tRNA. So the tRNA, I guess they call it that because it kind of looks like a T, but it also is called that because it transfers amino acids to the ribosomes. So what we see here that's of relevance to us is this tRNA has an anticodon and an amino acid. So it bears those 2 things. The anticodon will be complementary to the codon of mRNA. So here we see CUU is on the mRNA. Well, what's the anticodon? C pairs with G, U pairs with A, U pairs with A. So that's how the ribosome knows which amino acid to bring in, because the amino acid will be on the tRNA that has this anticodon specific for each codon. So if I were to do the next one, let's see. So the first one was CUU. CUU is our next codon on mRNA. So the next tRNA that would be brought in would be the same as the first one. We would find another tRNA that also has the same amino acid. In this case, by the way, it says it's leucine, but you don't need to know that. But if the codon was different, let's back up. GUC. The anticodon would be CAG. And it would bear another amino acid, a different one. For example, some just I can rattle off might be isoleucine, we have phenylalanine, alanine, valine, proline, glutamic acid, histidine, but again, y'all don't need to know all of those. So now let's look at everything together. So we see our ribosome, the large and small subunit with the mRNA threaded into it, and something that's important is that the codon that we're going to start with is always AUG. Now there may very well have been several codons listed before the AUG, but the ribosome's not going to start until this AUG is threaded into it. All right, so say that the AUG was here. That was a really terrible drawing, I'm sorry. There could have been other codons here. It's just going to ignore those because it has to find an AUG first. Hence why we call it the start codon. All right. So again, this is showing you a somewhat similar picture of initiation specifically. We see the small subunit, we see our mRNA, we see the start codon is present. There's some stuff before it, but again, the ribosome ignored it because it's looking for that AUG. We see the large subunit nestled right on top, and here we see a transfer RNA that has the correct anticodon for the codon. Codon on mRNA is AUG. What pairs with A? U. What pairs with U? A. What pairs with G? C. And it brought in the correct amino acid. This is called methionine. So that's initiation. Translation, elongation, is going to elongate the chain of amino acids that we are producing, which is what a protein is. So this is showing you, we started off with our small subunit, we have our codon here, we have our tRNA molecule here with the anticodon and amino acid, and then we see the large subunit gets sandwiched on. Now that was initiation, but now what we're seeing here is something new. We see that the A site has now brought in a new tRNA molecule that is complementary to the next codon. So AUG was complementary to UAC. UUC is complementary to AAG. And that is going to bring in this amino acid, which is labeled PHE, which stands for phenylalanine. So now let's compare this image to this one. What we're seeing is the P site has the green tRNA, and the A site has the blue. Compare that here. Now the P site has the blue tRNA, and the green tRNA is in the E site. So what happened here is the ribosome shifted down one codon so that the A site, or what was in the A site, is now in the P site. What was in the P site is now in the E site. And what we can see here is these two amino acids have now been covalently bound together. So this tRNA will now exit, hence the name E site, the ribosome. And then now the A site is empty and can bring in the next tRNA, which will have the correct anticodon for the codon on mRNA, as well as its associated amino acid. And this process will repeat, almost like an assembly line, as the ribosome shifts down 3 nucleotides, which is one codon at a time. So here we're seeing elongation, more steps of it. So this is showing you several steps in. We see various amino acids here that have been covalently linked, and at present, valine, that tRNA, is inside of the P site. Then we have this tRNA going to fill the A site, as seen here. Then this amino acid strand will covalently be linked to the next amino acid, as seen here. And then as that happens, this all will shift down one codon. So as we can see now in this third picture, this tRNA went to the E site and left. And what was in the A site is now in the P site in this next image. And that leaves the A site open for the next amino acid tRNA complex to come through. Now eventually we're going to read a stop codon. There's 3 possible stop codons, UAA, UAG, and UGA. Those 3 codons do not code for an amino acid, but it codes instead for release factors that will tell the ribosome to stop. Okay, sorry, I thought there was a video next. I'm going to play the video now. We'll come back to those other 2 slides.
How is the information in the mature messenger RNA strand translated into a protein? The nitrogenous bases are grouped into 3 letter codes called codons. The genetic code includes 64 codons. Most codons code for specific amino acids. There are 4 special codons, 1 that codes for start and 3 that code for stop. Translation begins with the messenger RNA strand binding to the small ribosomal subunit upstream of the start codon. Each amino acid is brought to the ribosome by a specific transfer RNA molecule. The type of amino acid is determined by the anticodon sequence of the transfer RNA. Complementary base pairing occurs between the codon of the messenger RNA and the anticodon of the transfer RNA. After the initiator transfer RNA molecule binds to the start codon, the large ribosomal subunit binds to form the translation complex, and initiation is complete. In the large ribosomal subunit, there are 3 distinct regions called the E, P, and A sites. During elongation, individual amino acids are brought to the messenger RNA strand by a transfer RNA molecule through complementary base pairing of the codons and anticodons. Each anticodon of a transfer RNA molecule corresponds to a particular amino acid. A charged transfer RNA molecule binds to the A site, and a peptide bond forms between its amino acid and the one attached to the transfer RNA molecule at the P site. The complex slides down 1 codon to the right, where the now uncharged transfer RNA molecule exits from the E site, and the A site is open to accept the next transfer RNA molecule. Elongation will continue until a stop codon is reached. A release factor binds to the A site at a stop codon, and the polypeptide is released from the transfer RNA in the P site. The entire complex dissociates and can reassemble to begin the process again at initiation.
Alright, so now jumping back to where we were. So as you saw in the video and as we've been describing, this ribosome is going to bring in the correct tRNAs based on what the codon sequence is that will result in a chain of amino acids. So there are 64 different triplet codes that are possible with three letters, and there's only 20 amino acids, so that's kind of interesting. That means that we must have some redundancy in our genetic code, and we do. Some amino acids are represented by several different codons, so we can have multiple codons that code for the same amino acid. And this lends a hand to our wobble effect that we describe in the genetic code. Basically, what it means is that the last nucleotide is not super important when it comes to determining what amino acid will be added. It's the first 2 nucleotides that are really required to be correct to encode the correct amino acid. Now, what's so neat about that is that means that mutations in our DNA can happen without ultimately altering protein structure. And that's important because protein structure is relevant to its shape, and if it doesn't have the right structure, it doesn't function. So pretty neat effect. Alright, so let's look at our genetic code table. So you definitely don't need to memorize this, but I would just want to show you how to work it. So if I were to ever give you a mRNA transcript, let's say AUG, G, 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 AGA, and we could keep going, but we'll just do 3. You should be able to look at this table and tell me what the amino acid sequence would be. So AUG is our first codon. The way we read this table is we go to the first base position and find the A's. That's here. The second base position, it says it's U, so it must be in this column. The third base position is a G. Well, we already said it must be in this column in this row, So now we find AUG, and it is the start codon, which is methionine. Now GGG, same process. First base, G, must be in this column, or row, excuse me. G over here, must be in that column. And going down, connecting our imaginary lines here, we see GGG, so that's glycine. I'm going to write GLY for short. And then finally, AGA, must be in this row, must be in this column, go to where they meet, and look for AGA. And it is arginine, ARG. So in reality, a protein is going to be tens of thousands of amino acids long, and it will fold up into a very specific three-dimensional shape. Okay, so all in all, this is just another example. We see the mRNA listed here. GUA is the first codon. It codes for valine, and you would find that out using your genetic code. Second codon was cysteine, and we know that because we looked up the codon on this codon chart, and then repeat for each one, till eventually we have this amino acid chain. That amino acid chain folds up three-dimensionally into a protein. Alright, so moving forward. On second thought, I just paused this and restarted it, but what I'm going to do is make this into 2 videos. So I feel like that's a lot to chew on, so this will be part 1, and come back and watch part 2 once it's posted.